Now, these chromosomes, so we still have a diploid cell. That dotted line is just showing you the middle. The next step then, we would actually have to separate homologous chromosomes. So we're going to separate them. So now we actually have taken matching chromosomes. Remember, this one has all the same characteristics as this one. So now we've separated them to opposite sides of the cell. And now we end up with two cells. Now, if you think about it, are these cells still diploid? Do they have two sets, one from the mom and one from the dad? No. These cells are now haploid. We have half the number. We started with 4 originally, and now we're down to 2. We have 2 chromosomes in each cell. Yes, they are duplicated chromosomes, but still, they're just chromosomes. Now, what's going to happen next is we still need to divide the DNA. Because if you think about it, if we were to separate it like this, we would end up with a cell that has 4 chromosomes. And we don't want that. We need a cell that has 2. We need a haploid cell. So our next step is we actually have to separate these sisters. So this is very similar to mitosis. At this point, the sister chromatids are all going to line up along the middle of the cells. And then we pull apart the sister chromatids. And then we end up with 4 cells. And each cell has only 2 chromosomes. We now have haploid cells. Now these are gametes or sex cells, and in this case they could represent sperm. Eggs are a different story, and I'll have a video up probably tomorrow about how eggs are formed. But anyway, so that is meiosis.